Dear students, Assalamu alaikum. This video is the continuation of my previous lecture videos on enzymes. Today, I shall discuss about the remaining parts of enzymes. Enzymes are known as simple enzyme or apoenzyme when their functional forms consist only of proteins with tertiary structure. However, many enzymes require small molecules or metal ions called cofactors to catalyze reactions properly. When the cofactor is a small organic molecule, it is known as coenzyme. Coenzymes are non-protein organic molecules that are mostly derivatives of vitamins soluble in water. If an enzyme requires a cofactor, neither the protein structure nor the cofactor alone has catalytic activity. Many enzymes must contain a metal ion to carry out their catalytic activity. The metal ions are bonded to one or more of the amino acid side chains. The metal ions from the minerals that we obtained from foods in our diet have various functions in catalysis. Ions such as ferrous and copper are used by oxidases because they lose or gain electrons in oxidation or reduction reactions. Other metal ions such as zinc stabilize the amino acid side chains during hydrolysis reactions. Many enzymes require cofactors to function properly. Cofactors can be considered helper molecules that assist enzymes in their action. They are able to assist in performing certain necessary reactions the enzyme cannot perform alone. Many cofactors sit in the enzyme active site and make the active site suitable for the substrate molecule. After that, the substrate molecule can easily bind in the active site of the enzyme and can form an enzyme substrate complex, which is necessary for the completion of the reaction. An inactive enzyme without the cofactor is called an apoenzyme, while the complete enzyme with cofactor is called a holoenzyme. Vitamins are organic molecules that are essential for normal health and growth. They are required in trace amounts and must be obtained from the diet because they are not synthesized in the body. Vitamins are classified into two groups by solubility: water-soluble vitamins and fat-soluble vitamins. Water-soluble vitamins have polar groups such as hydroxyl group and carboxylic group, which make them soluble in the aqueous environment of the cells. Most of the water-soluble vitamins are not stored in the body and excess amounts are eliminated in the urine each day. Therefore, the water-soluble vitamins must be in the foods for our daily diets. Because many water-soluble vitamins are easily destroyed by heat, oxygen, and ultraviolet light. That's why care must be taken in food preparation, processing, and storage. The water-soluble vitamins are required by many enzymes cofactors to carry out certain aspects of catalytic action. The coenzymes do not remain bonded to a particular enzyme but are used over and over again by different enzymes to facilitate an enzyme-catalyzed reaction. Thus, Only small amounts of coenzymes are required in the cells. Fat-soluble vitamins have nonpolar groups which make them soluble in the fat or lipid components of the body such as fat deposits and in cell membranes. The fat-soluble vitamins A, D, E and K are not involved as coenzymes, but they are important in many processes such as vision, formation of bone, protection from oxidation, and proper blood clotting. Because the fat-soluble vitamins are stored in the body and not eliminated, there is a big possibility of toxicity if taken too much either accidentally or unintentionally. Enzymes can be regulated by other molecules that either increase or reduce their activity. Molecules that increase the activity of an enzyme are called activators, while molecules that decrease the activity of an enzyme are called inhibitors. Enzyme can be regulated by feedback control proenzymes, allosterism, protein modification, and isoenzyme. Enzymes are often regulated by environmental conditions. Feedback control is an enzyme regulation process in which formation of a product inhibits an earlier reaction in the sequence. The reaction product of one enzyme may control the activity of another by competitive or non-competitive or some other type of inhibition. In the reaction, we can see that D is produced after a sequential enzymatic reaction, which may inhibit the activity of enzyme E1 by feedback inhibition. Some enzymes are manufactured by the body in an inactive form. To take them active, a small part of their polypeptide chain must be removed. These inactive forms of enzymes are called proenzymes or zymogens. After excess polypeptide chain is removed, the enzyme becomes active. For example, trypsin, an important catalyst for the digestion of the proteins, is manufactured as the inactive molecule trypsinogen, which is a zymogen. When a fragment containing 6 amino acid residues is removed from the N-terminal end of trypsinogen, the molecule becomes a fully active trypsin molecule. Sometimes regulation takes place by means of an event that occurs in another site other than the active site, but that eventually affects the active site. This type of interaction is called allosterism, and any enzyme regulated by this mechanism is called an allosteric enzyme. If a substance binds non-covalently and reversibly to a site other than the active site, it may affect the enzyme in either of two ways. It may inhibit enzyme action, which is known as negative modulation, or it may stimulate the enzyme action, which is known as positive modulation. The activity of an enzyme may also be controlled by protein modification. The modification is usually a change in the primary structure, typically by addition of a functional group covalently bound to the apoenzyme. For example, the active enzyme pyruvate kinase becomes inactive when it is phosphorylated. When the activity of pyruvate kinase is not needed, it is phosphorylated to inactive pyruvate kinase phosphate by a protein kinase using ATP as substrate as well as a source of energy. When the system wants to turn on pyruvate kinase activity, the phosphate group is removed by another enzyme, phosphatase, which renders pyruvate kinase active. Another type of regulation of enzyme activity occurs when the same enzyme appears in different forms in different tissues. These different forms of the same enzyme are called isoenzymes. Isoenzymes perform the same function but have different combinations of subunits and thus different quaternary structures. Most enzymes are confined within the cells of the body. However, small amounts of them can also be found in body fluids such as blood and urine. The level of enzyme activity in these fluids can easily be monitored. This information can prove extremely useful. Abnormal activity of particular enzymes, that is, if their amount either high or low in various body fluids, signals either the onset of certain diseases or their progression. So, by monitoring the level of enzyme activity in our body fluids, we can diagnose certain disease and their progression. That's all for today. Hope all of you enjoyed my lecture videos. If you like my lecture videos, then don't forget to click the like button. You can also share these videos to your friends and can also put your comments. Lastly, don't forget to subscribe my channel. Your subscription will encourage me to make more lecture videos for you all. Thank you.